us today, and thank you for your continued leadership, both prior to your time at FDA and over the last many months that you've been hard at work there. And thanks to all of your colleagues that are here today, led by Dr. Pazner and his team. You'll hear from many of them throughout the day today. So in order to get started, I'd like to invite the first panel to go ahead and join us on stage this morning. For those of you that haven't been a part of this meeting before, I want to highlight the work that has gone into this. Each of the sessions that will be presenting their work today have proceeded with several months of deliberations to prepare the white papers that have been distributed in advance of today's meeting. While our experts today will be presenting the ideas and creative approaches to each of the three innovative topics today, we really want to hear your feedback. From our standpoint, we're excited about these discussions. I think we have three terrific topics this year, and we're already starting to think, how do we implement? How do we move some of these forward, and what venues can we facilitate in order to make that happen? Before I turn things over to Dr. Stewart, I want to thank our team and friends here who has helped coordinate all of this, led by Mark Stewart, Diana Marino, and Laura Lassiter, who have helped facilitate these panels over the last couple of months. So we look forward to a great discussion today. There should be time built in for each of these for plenty of questions and ideas to move forward. So thanks so much for all of your work. We look forward to hearing from you.
All right, thank you everyone. I'm Liz Stewart from Johns Hopkins, and I'm happy to orient you to this first session with the sort of one of the three white papers. Let's see if I can get this. We'll have the panelists introduce themselves in a minute. So this is the title for the session, Augmenting Randomized Confirmatory Trials for Breakthrough Therapies with Historical Clinical Trial Data, Landscape Review, and Topic Introduction. What I'm going to do is take just a couple minutes to orient you to, I can put this down so that you can sort of see me over the podium. I'm going to orient you to the session. I'm just going to give sort of the big picture, because from that title, it's a little bit of, might be a little unclear, just given the length. So the big picture here is, as we just heard, there's a need for innovative designs to speed getting effective treatments out into the public. And one aspect of that might be the need for non-experimental designs. But then the question that arises is, how do we know when we can trust the results from those studies? There's a growing literature to try to answer that question, and that's part of what we're going to contribute to today. And in particular, we're going to discuss two case studies that examine this, and also put that into a broader conversation about the possibilities and the issues that come up. So let me just say a couple more words about the need for non-experimental studies. So I do a lot of work in non-experimental study design, and there's many questions that we can't answer using a typical randomized design. Examples of these might be the effects of approved treatments once they're already in real-world use. Sort of once something is already out, we might still be interested in safety or long-term effectiveness. In fact, as we just heard from Anna, she gave a couple examples of that. Another example are these ethical scenarios where it might be unethical to deny some patients access to a new treatment. And then finally, I do a lot of work in public health where we might be interested in the effects of carcinogens or other factors where we're not going to expose people to that. We just have a scenario where some people have exposure to it and others don't. So in these cases, we might be left with a non-experimental study where we can't control who received the intervention or sort of comparison or control condition. There's lots of designs for this. I teach term-long, semester-long courses on this. Today we're going to focus on one particular design, that of a comparison group design. And so the challenge here is the sort of fundamental challenge of non-experimental studies is that we're worried about what's called confounding. The fact that people that get one treatment or exposure may be quite different from those that get some comparison. The people that get exposed to some carcinogen might be different in other ways from people that don't. So there's a variety of statistical methods that can be used to deal with this. But to interpret the difference, we're going to hear about one particular approach today. The two case studies both use something called propensity scores, which we'll hear more about. So in those designs, you can take advantage, you can sort of try to deal with differences due to observed characteristics, differences in age, gender, health status, things that we can observe. But the problem is that we might still be worried about unobserved differences between the people in the different exposure groups. And so then the question really is, when can we trust non-experimental results? And can we build a body of evidence to help us understand that? So that's really, again, the topic of this session. We're going to hear about 2 case studies seeing whether historical comparison data, and in particular, data from historical clinical trials, from the control arms of historical trials, and whether those can yield accurate effect estimates when we compare them to a more recent clinical trial exposure group, treatment group. And in particular, what we're going to be looking at today is whether the average outcome seen in the control condition of a current trial matches the outcome seen in the historical data, from a historical control arm. And you'll hear more about that. I'm giving a 50,000-foot view right now. And this, I would want to frame this as this is 2 case studies, but I hope that this is the beginning of a start at developing a whole body of literature on this topic. So moving from an N of 2 to a larger sample will help us really start to understand trends and when we can trust these sorts of studies. And just on that note, I want to just quickly allude to another area that is developing a very large body of literature on this topic. And there's this whole field, and this is essentially what we're doing today is an example of this, what's called a within-study comparison. So this idea where you sort of pretend you didn't have a trial, you try to see if you can replicate a trial result using non-experimental data, and you use that to learn about what requirements are needed in order to be able to trust the non-experimental study design. This is sometimes called design replication studies or within-study comparisons. And in the social sciences, the social behavioral sciences, Tom Cook, who's at George Washington University currently, has assembled 100 of these. So he has assembled 100 of these, and he now has written papers where he can examine sort of what are the properties of these studies that lead to accurate non-experimental effect estimates. And I sort of say that just as perhaps inspiration for the field of cancer research, that the more you can build this sort of body of evidence, the more you can learn to sort of understand, well, when can we trust one of these historical control studies, and when really do we need to do a concurrent randomized trial? So today, again, we're starting this conversation with these 2 case studies to help us try to understand how much of the findings from the social sciences might carry over, or what else do we need to learn for cancer research. This is the plan for the session. We'll do brief introductions of the panel, then 2 case studies for about 10 minutes each, panel discussion, we might shorten it to more like 30 minutes, and then we'll have some time for open question from you in the audience. So why don't we start with just quickly, each panelist can go down and introduce themselves, with sort of who they are and their interest or relevant expertise for this area.
Hi everybody, I'm Ruthie Davey. I am Vice President of Data Science at Medidata. I'm a statistician by training and held previous roles at the FDA in statistics or managerial levels in the Office of Biostatistics. I'm interested in this topic today. Medidata has an archive of historical clinical trials available for this kind of work, and this is fueling a lot of our interest today.
Hi everyone, my name is Pallavi Mashrakoyani. I'm a team leader in the Division of Biometrics 5, which is in the Office of Biostatistics. We are the division that supports the review of oncology and hematology products at the FDA.
Hi, I'm Andrea Ferris. I'm the President and CEO of Longevity Foundation. I am not a biostatistician. I actually had to Google propensity score to try to figure out what the hell it meant in English. But I'm here and our interest has been more from a patient perspective, which is really to understand how do we streamline clinical trials, make them more efficient, and in this era of big data, how do we utilize that to help get drugs to market faster, as well as to enable patients to go on to an active arm.
Hi everyone, my name is Judy Self. I'm a medical reviewer in the Office of Hematology and Oncology Products, and I focus primarily on rare diseases, brain tumors, and pediatric diseases. This is an area where, as we all know, it can be often difficult to do randomized controlled trials due to population sizes. That's really the start of my interest in this area. I also had to Google propensity scores before our first meeting, just so that I wouldn't feel like a complete nincompoop on teleconferences. But I'm very excited for us to share the work that's been done.
Good morning. I'm Antoine Hubert. I'm currently the global head for R&D oncology for Daiichi Sankyo, so probably the only industry rep on the pharma side. I've been blessed by 30 years of drug development globally. Initially, Taxotere, which is a drug that you may know of, and more recently, drugs like Tagrisso or the first PARP inhibitor, olaparib. And now currently at Daiichi Sankyo, we have actually 3 drugs which are breakthrough designated, including antibody drug conjugate, which deliver extremely high response rate. Out of this recent experience, I came to realize that we have an obligation to actually use these drugs where the evidence is compelling to actually advance the science of drug development so that we can withstand the uncertainty that Elizabeth was describing in the manner which is useful. So that's where our interest is my company's commitment to support this effort, because we have enough science so that we can actually absorb the uncertainty and progress the science for the benefit of patients.
Hi, I'm Rajesh Sridhara. I'm the Division Director of the Biometrics 5 that Valerie was talking about. And our division supports all of the oncology and hematology products. We are a group of about 40 PhD statisticians who are totally dedicated to oncology and hematology. I think I'll stop there. I've been with the FDA for 19 years, close to 19 years.
Great, thank you. So now welcome Ruthie Davey to talk us through the first case study. It's my pleasure to be here today to talk with you about something called synthetic control arms. This is the beginning of a conversation about how we might utilize historical clinical trials data to address some of the challenges we're facing in conducting trials with concurrent control arms in difficult-to-study indications, such as often the case with breakthrough therapies. The work that I will share with you today is a result of a unique collaboration that Friends has facilitated. The SCA working group includes members from industry, academia, as well as the FDA. We're excited to be working with this talented group that you see on the stage today, and they're all very modest, saying that they had to Google topics before they came here. They each have their own strengths and have brought a lot to this group. And we look forward to continuing this research even after the presentation today. So I will begin by describing the challenge that we face. As I'm sure you know, many products with breakthrough therapy designation are approved through the accelerated approval pathway. This means there is early access to these products for patients, but it also may compromise the clinical equipoise needed to conduct confirmatory randomized controlled trials. Patients may not be willing to enroll in a trial where there's the possibility of being randomly assigned to a perceived inadequate standard of care, or they may discontinue or be incompliant with trials that are ongoing when an accelerated approval comes on the market. The BRAVO study in BRCA-positive cancer is an example of this. According to a press release by the sponsor of this trial, there was an unusually high rate of censoring in the control arm, likely associated with the availability of a PARP inhibitor. The study was terminated early since it was unlikely to produce useful data. Sutton and Malley was studied for gastrointestinal stromal tumor and demonstrated a large effect on progression-free survival. However, after the PFS analysis, patients were able to receive the investigational product, and 84% of the placebo patients did so. At the subsequent analysis of overall survival, no effect was demonstrated. Both of these cases illustrate examples where the randomized controlled trial as a research tool is breaking down. We believe better statistical methods are needed to address these challenges. We propose today that synthetic control arms may be a solution to this problem. There is still lots of work to do, but we'll show you the results of a case study that support this possibility. So, what is a synthetic control arm? A synthetic control arm is made up of historical clinical trials data from trials conducted in the same indication. We take historical patients who meet the eligibility criteria for the current day trial and were assigned to receive the same standard of care,
And we select them based on their baseline characteristics to match the baseline characteristics of the experimentally treated subjects. In this way, we create something that looks a lot like a randomized control for a setting where randomization is problematic. I'll illustrate what I mean. Suppose we have a set of current day experimentally treated patients for whom it has not been possible to enroll a randomized control arm. Each of the patients in this figure are represented by a green dot. And without a control arm, this data is very difficult to interpret. And so we aim to create a control arm from historical clinical trials data. Here, the historical trials are represented by circles and the historical patients by dots. These trials come from a single indication, the same indication for which the green patients are being studied. These studies enrolled under similar but not identical eligibility criteria to the green patients. We start by curating and standardizing the historical data. This removes the boundaries of the trials and allows a combined analysis. We then apply the eligibility criteria used for the green patients to the historical patients. Some patients will satisfy them and some will not. In this graphic, the yellow patients are the patients that did not satisfy these eligibility criteria, and they are removed. Now we have two sets of patients who have all met identical eligibility criteria. However, the patients are not all identical. They differ in terms of their baseline characteristics, things like age, or ECOG score, or disease severity. And in order to have a fair comparison, we want to be sure that those things are balanced in the experimentally treated patients and in the historical control. And so we now use the propensity score method approach to create balance in these characteristics. We select patients from the historical pool with the appropriate baseline characteristics to match the patients in the green trial. And you'll notice that the intensity of the colors is now lined up. We have a good match between the historical data and the current day experimentally treated patients. This allows the apples-to-apples comparison that we need. So, what would have looked like this with an uncontrolled experimentally treated set of patients now looks like this. We have the context to understand whether the experimental product is offering a positive effect or not. But to believe that we've provided the correct context with the synthetic control arm, you have to believe that this synthetic control has matched the result of what a randomized control would have demonstrated if it had been possible to do the randomized control. That is, we would like to see as close of alignment as possible between a synthetic control arm and the randomized control arm. And this is the objective of our case study. For our case study, we start with a completed randomized controlled trial. We create the synthetic control arm from the historical data, much the way I've described. But instead of matching to the experimentally treated patients in the target trial, we match to the randomized control of that trial. Our goal is to end with a figure like this that shows a replication of the overall survival using the synthetic control arm as what is seen in the randomized control arm. So let's get to our case study. This case study is carried out in non-small cell lung cancer. We'll start by building the SCA. The case study uses data from multiple historical clinical trials. These trials are available through Project DataSphere and the Metadata Enterprise Data Store. These trials were either open label or blinded. They were phase 2 and phase 3 trials. All trials were multinational and were conducted during the time period from 2004 to 2013. All trials measured overall survival as a key endpoint. All patients included in this case study meet the eligibility criteria you see listed here, as well as were assigned to the control arm of those historical trials and received treatment with docetaxel. This slide describes the implementation of the pre-specified propensity score matching. We use the baseline characteristics listed here to develop a propensity score for each patient. We can think of this score as a summarization of all of these baseline characteristics and the impact that this has on the likelihood eligible to receive the treatment under study. On the bottom of this figure, we see the propensity scores mapped for the historical pool of patients available for the SCA. And on the top, we have the same for the randomized control from the trial. We can see that there is a considerable amount of overlap in the distribution, but it's not complete. There are some areas of propensity scores that exist in the randomized control that do not exist in the historical pool. The green subjects, the green dots, represent subjects who are appropriately matched to a historical patient. That is, there's a historical patient that has a propensity score that's sufficiently similar to call that a match. The red and blue symbols, though, represent patients in the randomized control from the target trial who are not matched in historical data. These patients are excluded from further analysis. Now, for those of you who are accustomed to clinical trials, this may be a little unsettling. This thought of excluding patients from the analysis goes against the intent to treat principle and may lead you to think there could be an imbalance between the two groups. That would be true if this were a randomized controlled trial. But this is not a randomized controlled trial. Randomization has not been utilized in this setting, and removing patients from this group actually improves balance rather than threatens it. Okay, so we're going to move now to see how we did. We're going to look at the balance at baseline in the newly created SCA and the randomized control arm from the target trial. We'll start by looking at the propensity score. Remember, this is our overall measure and summarization of those baseline characteristics. On the bottom of this graph, you can see that all historical patients in the set of, using all historical patients and the randomized controlled trial, there is significant difference in the propensity scores. The historical pool generally has a lower propensity score than those in the randomized control from the target trial. This is before matching. This would not be an apples-to-apples comparison. After matching, though, we see that the distribution of propensity scores is well balanced between the two groups, and this would lead to an apples-to-apples comparison. As I mentioned, the propensity score is a function of the baseline characteristics, so what I'm going to show you on the next slide should really come as no surprise. Here we're looking at the balance between the groups using each baseline characteristic alone. This display is mostly for people who are used to clinical trials data. We take each characteristic and look on its original scale at whether it's imbalanced between the two groups. We do see some significant imbalances in the proportion of patients with prior surgery, also the proportion of patients with stage 3 disease, with squamous histology, and there are imbalances in smoking status. After matching, though, we see that nearly every characteristic is well balanced, even those 4 that we mentioned that were imbalanced prior to matching. Again, this supports the assertion that we have an apples-to-apples comparison. Now, if you're not a clinical trialist and you're more like an epidemiologist, this slide is for you. This is the same information showing that we have reduced the absolute standardized differences in the baseline characteristics. In this graph, each line represents a single baseline characteristic, and the sort of downward trend that you see is a reduction in those differences after matching. Before matching, there's considerable imbalance, and after matching, the absolute standardized differences are reduced to a level that is, for all but one, is below our pre-specified threshold of 0.1. This is a commonly used threshold for determining that the differences are negligible and that it's a fair comparison between the two groups. All 3 of these slides illustrate that the propensity score matching has been largely successful, and we can move to a comparison between the groups. Okay, so now I'm about to show you the most important finding from the case study. So a few of you may be getting tired or overwhelmed by the statistical material. But I just ask you to rejoin me with renewed interest for 2 minutes. Okay, so here it is. This is the overall survival outcome for the historical pool of the data versus the randomized controlled trial before matching. And looking at these Kaplan-Meier curves, we can see that there are differences in overall survival. This is supported by the log-rank test with a significant p-value and a hazard ratio and confidence interval that lie completely above 1. This should be concerning to the clinical trialists in this room. This suggests that historical comparisons that are naive without any matching procedures could be leading us to the wrong conclusions. After matching, though, we see significant overlap of the survival curves, suggesting that the SCA has successfully replicated the result of the randomized control. There's numerous places where the curves cross or intersect, and it's also supported by the log-rank p-value being not significant, the hazard ratio and confidence interval being centered at 1. This opens the possibility that SCA could be used in future trials to augment or replace a randomized control when a randomized control is difficult or problematic. So in summary, this case study in non-small cell lung cancer the SCA has successfully replicated the results of the randomized controlled trial. We think this is a very important step in mitigating the challenges we see with conducting concurrent control arms for these difficult-to-study indications. We do believe there's much future work that's needed. This working group in particular has plans for an assessment of whether the treatment effect can be replicated with the SCA. That is to say, the difference between SCA and experimental treatment patients, does that match the difference between a randomized control and the experimentally treated patients? We also believe it's important to try to tweak the matching approaches that we're using in order to not exclude such a large portion due to mismatching. And then, of course, we believe it's necessary to expand this to additional indications. But overall, we're very pleased with the outcome of this case study, and we think it is very encouraging for the possible use of historical clinical trials and synthetic control arms going forward for these difficult-to-study indications. I want to thank you for being here today and for your attention to the presentation. I also want to thank Friends for including us in this work and allowing us to be here to talk to you today. It's now my pleasure to introduce Pallavi, who's going to share a second case study.
Thank you, Ruby, and thank you, Friends, for allowing us to present the second case study here today. So today I'm going to present a case study using external controls, and in a few minutes I'll discuss why that's a little different from what Ruby discussed as synthetic control arm. And in this particular example, we use a case study of melanoma data. So, typical disclosure slide. So, not to harp on what we've already heard, but there are definitely, I think the evidence still exists that randomized clinical trials are still the gold standard for identifying treatment effect. But there are times where randomized design isn't possible. This may be due to disease characteristics, it may be due to population characteristics, but regardless, when that's the case, we may look at data from trial designs such as single-arm trials to identify some estimation of benefit to a patient population or estimation that there is some sort of advantage to treating them. However, if a randomized controlled trial is infeasible or a control arm is not feasible, we can consider using an external control arm as an option for estimating comparative treatment effect. There are a lot of advantages and disadvantages to this, but we could consider external controls for identifying the natural history of the disease, for use in identifying the contribution of components. This could be a trial where you may have a combination arm as treatment and you want to compare that to a standard of care, but would like to understand what the contribution is of each of the treatments used in combination. Or it could be used to establish the efficacy from prior trials. For example, as I just mentioned, if you were to consider using a single-arm trial to identify some activity of a treatment, you could compare that activity to perhaps what you found in prior clinical trials for other treatments that are used in that similar population, or same population. The source of the data that are used for controls would determine the potential use of those controls. There are pros and cons of each type of data. There is data that can come from a registry or even electronic health records, and then there may also be data that can be used for prior clinical trials, such as what Ruthie presented in her presentation of the case study in lung cancer. We consider a case study of using an external control for identifying an effect in melanoma, where we look at two clinical trials. The first was a comparison of vemurafenib versus dacarbazine. This trial initially started enrolling patients in 2010 and had primary endpoints of PFS as assessed by an independent review committee and OS as co-primary endpoints. This trial went on for about 5 years, so the final patient was enrolled within that first year, but the patients were followed through 2015. The second trial was a design looking at cobimetinib plus vemurafenib versus vemurafenib alone. The primary endpoint in that trial was PFS also, but that wasn't as assessed by investigator. In that trial, the first patient was enrolled in 2013, so there was a lag of about 3 years between the enrollment of the first patient in trial 1 and the first patient in trial 2. So our question that motivated this case study was that could the data from the first trial, of the vemurafenib arm of the first trial, be used as an external control for the control arm of trial 2? Meaning, could we compare the external control arm of vemurafenib from the first trial to the experimental arm of cobimetinib versus vemurafenib of trial 2? So in order to compare these two trials, and really in order to compare data from any two sources, the first thing we need to identify is whether the data is compatible. That means, do the inclusion and exclusion criteria match? Is there consistency of the endpoint measurement? And can we identify methods that we may be able to use to reduce any bias in estimating treatment effect? In this case, comparing the trial of vemurafenib versus the trial for cobimetinib plus vemurafenib, the inclusion and inclusion criteria were nearly identical. That really was very advantageous for this case study and kind of why we chose it. We did have a harder time with the endpoint measurements because in one trial, the PFS was assessed by independent review committee, and in the second trial, the PFS was assessed by investigator. However, in the first trial, they also assessed PFS by investigator, so we chose to use that secondary endpoint of PFS by investigator as the identification of the endpoint that would be used for identification for treatment effect. And we also looked at overall survival. However, with the two trials having different primary and secondary endpoints, that also affects things like follow-up of the endpoints, and that might also affect the comparison of those endpoints when looking across trials. Finally, in this case, we were able to identify enough information that was common between the two trials to use a method called propensity scores and use those in particular for matching across the two trials in order to reduce bias when looking at efficacy across the two trials and trying to identify a comparative treatment effect. In this case, we used a logistic regression model for treatment to calculate the propensity score using baseline covariates such as age, sex, region, and so forth as listed at the bottom of this. When we compare the two populations of the trials before matching, what we see is that they're actually fairly similar. And the reason for this, I think, is because these are both targeted treatments in a specific population. And we see that the inclusion-exclusion criteria also helped to make sure that the trials look pretty similar. There are a few differences. The region across trials is slightly different, and there are more patients enrolled in North America in Trial 1 than in Trial 2, and same for Europe. In Trial 2, there are more patients in Trial 2 than in Trial 1. And also we see that prior adjuvant therapy is more common in Trial 1 than in Trial 2. However, overall, the patients look fairly comparable even before we use any methods for eliminating bias or reducing bias. Also, when we look at the initial efficacy results across trials without any matching or any other methods, we see that while PFS by investigator is fairly similar across the two control arms, the vemurafenib external control arm and the vemurafenib concurrent control arm, the overall survival is actually fairly different across the two trials. With the vemurafenib external control arm, so the vemurafenib arm from the first trial, having a lower median survival, and that's the lower blue curve in that first plot, compared to the vemurafenib concurrent control arm, which has a higher median survival, and it's the darker blue line or the higher blue line in that first plot. So the question is, if we are able to match our populations, will we see more similar results in the control arms, and what will that mean for our comparative treatment effect estimation? So, as you may have expected, when looking at PFS, we see fairly consistent results regardless of the methods that we use. The first set of columns in this table represents the results from the original trial of vemurafenib versus cobimetinib plus vemurafenib. The second two columns uses all of the external controls, so all of the patients from the original trial, or the first trial rather, of vemurafenib, compared to the experimental arm of cobimetinib plus vemurafenib. And the last two columns use the matched analysis, or I show the results of the matched analysis. So those are the patients who are matched from the first arm, trial arm of vemurafenib, the external control, to the experimental arm of cobimetinib plus vemurafenib. So across PFS, we see that the hazard ratios stay fairly consistent. We start with a hazard ratio of 0.51 in the original trial, and we stay fairly close to it across the analyses using the data from trial 1. However, when we look at overall survival, we see that the original trial, or trial 2, has a much higher median survival time for the control arm of vemurafenib of 17.4 than all of the external controls from trial 1, which give us a median survival time of 13.6. However, after matching, we do see that that median survival time does increase slightly to 15.4, and the hazard ratios going across also look more similar to what we see in the original trial. So if we were to use this data, if we were to use matched external control data, we might actually come to a similar conclusion to what we did in the original trial of trial 2, cobimetinib plus vemurafenib versus vemurafenib. When we look at the Kaplan-Meier curves for survival, we see a similar story. Things are looking a lot closer when we use matching, especially when comparing the two control arms, as well as when you look at the matched experimental arm compared to the original experimental arm. So this case study shows that there is some promise to using some external data. In this particular example, we used the data from one clinical trial to substitute for another clinical trial, which is different from what Ruthie presented, which would be to use several clinical trials together as a synthetic control arm. That may be more robust, but there are also challenges there because you have a lot of different trials with different inclusion-exclusion criteria that must be matched. In this particular example, we were able to use, to our benefit, the fact that the inclusion-exclusion criteria were very similar across these two trial arms. two trials. The enrolled patients were from a similar targeted population, and so we were able to find very similar patients to compare. Considerations for using external controls in general have been covered, but just to quickly go over them again, we need to be able to compare the endpoints. The temporality of the data is important. Even small lags may make a big difference. It's hard to tell whether the overall survival in the bemorafenib concurrent control arm was better due to new advances in medicine, or whether it was a slightly different patient population. That may be difficult to tell just from this single case study. Treatment, patient, disease, population, idiosyncrasies are always difficult to identify in studies like this, whether it's a single case study or whether you're using several studies to create a synthetic control arm, or if we look forward and do this again and again, we may have other issues that come up. So in the future, we'd like to do the same type of analysis in other disease areas and also try to replicate what was done by this working group by looking at several control arms or several arms from other clinical trials to create a synthetic control arm. Thank you very much.
Great, thank you both for the nice overviews, and of course everyone has the white paper if you want to see all the nitty-gritty details or read it on the train home or something. So we're going to now turn to about 30 minutes or so of panel discussion. As you heard earlier, we have a great diverse set of perspectives and expertise on the panel, so we want to make sure to take advantage of that. And first I'm just going to ask the panelists, especially those who didn't present a case study, to sort of provide just any reaction you have overall to the case studies and this general idea, especially within the context of the findings from these two. So maybe I'll start at the other end, actually, and ask Raji to start us off.
So, these were case studies where it seems like it can work, but so in other words, it is encouraging. However, we have to be cautious. Just last week in another meeting, one of them from Eli Lilly presented about oxaliplatin looking over a period of time. Initially, the response rates were lower, but later on it got higher, and when they examined, it was simply because they were able to give for longer time oxaliplatin in colorectal cancer. So as you go along, you learn and you may be able to manage the patients better, and so the duration of the treatment and the follow-up and everything could change. So we need to be aware of this temporal drifts or shifts that can happen in the control arm with respect to not only subsequent therapies that have come up, as well as the same therapy itself, whether you are able to deliver better. So, while this is encouraging, I would say, we have to examine a broader, spectrum of clinical trials, especially if it is targeted therapy, and if different assays are being used in different, trials, prior trials compared to now, in identifying the population, then do you really have the right population?
Thanks. Yeah, I think that the context of, also what you're saying is you really need to have a good understanding of the clinical context and have that clinical expertise to know, has the sort of standard of care treatment been changing? Can we really use them? And some of that isn't necessarily going to be observed in the data and has to be brought in through really content area expertise. Antoine.
Okay, thank you. And thank you for your point. I think that you use the word caution. I use the word caution for industry and for being on the pharma web. I think in pharma word will be worried. So I think that they're not. But we also say that this seems to work, and there is evidence that there's potential. Then I think we have to go back and really look at what is the primary solving. The primary solving is not our comfort level. The primary solving is very tangible. It's twofold. Number one is that we're randomizing patients for breakthrough therapy, meaning for therapies which have proven themselves to have high likelihood of having a hard clinical benefit. We're randomizing patients in great numbers for the purpose of randomized clinical trial. So essentially exposing patients to the inability of having a drug. So that's a real problem. And the second problem, which is also extremely tangible, is that in randomized clinical trial, prospective trial, again and again, when you have these magnitude effects and drugs which are accelerated approved, essentially you're confronted with a crossover which confound completely the ability to know anything. So on the one hand, we have to be cautious, on the other hand, we know nothing, and currently what we're doing is terrible. We just are looking at modeling for the purpose of payers and access across the world, the nice of the world, which are terrible methods, and it's more than enough. So I think that we need to be a little bolder, recognizing that it may work, but also we also have a duty to advance that science, especially if we have drugs which are great drugs, and they can withstand some uncertainty and advance the science and not only be cautious. So I'm far more optimistic than you are with all due respect.
Just because he asked that, although leave others to talk also. Even in randomized studies, we have done imbalanced allocations, so it doesn't have to be that you have to allocate one-to-one. And at the end of the day, by doing a randomized study, even a very small control arm study, you get a lot more information, especially currently in the landscape where the treatments are being approved at such a fast pace and science is changing so much that what's historical yesterday is truly historical and you may not be able to use them at all.
All agree, I'm just saying.
Judy.
So I had no idea what the results were going to look like. So I actually was waiting with bated breath to see the curves come out at the end. And I think I was surprised at how well matched it was. And I would echo what Raji said, that I think overall it's very encouraging. But again, my sort of propensity is that if you've got one success, is to try and do it again and again and again and make sure that it's not just a fluke. So I think we're looking forward to seeing a larger body of experience. I think also, even though this meeting is really about looking at, or the topic for this session is really about looking at augmenting or potentially replacing the control arm in randomized studies, I think there are a lot of other applications that this approach can be used for. So things like, to some extent, we do it a little bit already, looking at historical controls for hypothesis generation, looking at a combination of elements for treatment if you've got combination studies. So I think that there are definitely other applications to be used. The one slogan that I sort of thought about over and over again during all our teleconferences was reduce, reuse, recycle approach, which this sort of embodies that. So I think it's very helpful to think about trying to squeeze as much usefulness out of existing clinical trial data that we have if we can.
Andrew.
So for me, I thought it was actually very interesting and very, as you said, optimistic in terms of the results and so forth. There are a lot of these types of projects, endeavors, things going on right now out there that are demonstrating sort of the concordance of historic data, prospective data with control data, and which is fantastic. I guess my question is just, when does perfect become the enemy of good? And when is good enough? And when do we put sort of the stake in the ground to say, okay, the methodologies exist? I mean, you guys have all demonstrated from a statistical perspective that we can build it, and we have the data out there if we can figure out how to aggregate. And it's dirty data, which is a whole different issue of how do we then use that. And I think that there are sort of, in just hearing the case studies as well as the conversation, two pieces. There's sort of the group for which standard of care has not changed in many, many, many years, in which case historic data perhaps a little bit more concurrent, but historic data has the possibility of having utility there to demonstrate something, so you don't have to continue to randomize people when you pretty much know what's going to happen. And then there's the group of people for whom the standard of care is changing super fast. And so it's changing continuously, even when the trials are enrolling, still standard of care is changing. And so how do we use data to augment that and to help that and restructure EHRs and start gathering prospective data in a way that it will have real world utility? So So I think there are two different conversations, but both solving for a similar issue.
Great, yeah, I would agree, and I think, yeah, echoes nicely on Raji's points about, you know, sort of, we need to know about the context to know when we can trust these methods. I sort of sit often in social sciences and education, and there, the research has shown that, for example, you really need local controls. You need people sort of in the same labor market, let's say. The analogy here might be in some context you need time concurrent. You know, that sort of the localness is really more about time and clinical care.
But I think there's also a meeting in the middle. You know,
yes, well, again, it
statistical
depends,
perfection
exactly.
of knowing everything, and then there's what patient advocates like myself are pushing for, which is let's just put everybody on the app. But I understand the integrity of not doing that. Dr. Pazzer has schooled me in that very well. But there's validity to it, that you do need to be able to establish efficacy and safety, and part of that is the control. And I get that, but there's some place to meet in the middle between statistical perfection and the honest.
Pallavi, did you want to react any more to the broader context?
Sure. I think that it's something that I brought up in my very last slide, but I didn't really focus on it too much, which is what Raji mentioned, which is the temporality of the data. And I think I really agree with Andrea there that there are a lot of very fast-paced developments in oncology, but there are also groups of patients who have been taking the same treatment for years and years and years and years. And I think that Longevity before has done, this exercise with small cell, and I know that there have been groups of like esophageal cancer or other small groups of cancer patients who really haven't had quite as much innovation driving their treatment options. And so temporality may be subjective depending on the group of patients that you're considering using this type of data for. I think that another thing that I kind of mentioned again, and I think Ruby also mentioned, is the concept of augmenting clinical trial data. I think that's another very promising avenue for using these types of methods and these types of data, is that we don't always require one-to-one randomization. You could have a small concurrent control arm that's augmented with some historical control data, particularly data from previous clinical trials, because that does seem to be clear. We do feel more comfortable in exploring it and identifying treatment effect because we know that things have been measured in a certain way, and we know that it's been measured at a certain time point. So that's another option that is something that I think we could really think about in the future.
I think Antoine wants to react to that, and then we'll come back to Ruthie.
Thank you. Yeah. No, I don't want to throw to ourselves. I mean, we keep using the concept of replacing. I don't think we are ready to replace, but augmenting is a fantastic concept. I'm sorry. The point here is, what does augment mean? What is the purpose of augmenting? And this is where we really have to work with the stakeholders, both the science, the regulators not only in the US, but the regulators of the world. Because if we prospectively augment clinical trial, how will this be handled? How will the FDA accept not to have patient-level data, for instance? Because most of these methods essentially remove the patient-level data. How the sponsor will accept to handle this? What will be the meaning for the purpose of European agencies, the PMDA of Japan, the payers, and so on? These are also complete unknowns that unless we work together and define what will be the benefit of this, not only the advancement of science, but also allowing that. So augmenting is a great concept, except that it should not be a pure academic concept. It should be tangible in what would it mean from the review, from the labeling perspective, from the payers and access, and across the different regions of the world. So I just wanted to throw that dimension.
So I think Raji wants to add on to this point, so Ruth, did you mind waiting more?
Separate.
No, there are statistical methods and clinical trial designs which are there where we can augment and do some of this. If you, in places where, in disease areas where you can do a randomized study, you should be doing a randomized study. But if it's not possible, or it's a place where the standard of care has remained the same forever, and can we use that data to augment the concurrent control? Those are excellent places where you can look at that. I don't think that that's an impossible task to do. If we look at the spectrum of single-arm studies to randomized controlled studies, this is in between. It is certainly better than single-arm studies, which we are already accepting. So I don't, even though I'm throwing the caution there, what we are doing, we should be careful and we should be mindful of where we are getting to. It's not to say that we should not be using. This is certainly better, and among the sources of data where you can use case history, literature, or registry data, or real-world data, the data that's coming from a clinical trial is more curated already, and we have the strict eligibility criteria. We know exactly how they measured endpoints, which may not be there in other sources of data. So certainly using prior clinical trial data is better than others.
Let's let Ruthie react. I actually consider myself very fortunate to be sitting at this end because all I have to do is agree with what's being said to my left, which I do. I wholeheartedly agree with the statements Raji just made about sort of that continuum of evidence all the way from a single-arm trial to a randomized control, and this certainly being better than the single-arm trial, maybe approaching a randomized control, but that's yet to be seen with more work that we do. I do think, too, that the statements Antoine made about this being an approach that would be used for augmentation, not complete replacing of the randomized control, is a really great happy medium where we see a lot of the benefits for the patients, where more patients are getting assigned to the active arm. I know we do 2-to-1 randomization or maybe 3-to-1 randomization now, but we could do something even better than that, like 5-to-1 or 6-to-1 if we're replacing with historical data as well. So I think this is a really exciting time. Great, thank you all. I'm going to now turn maybe to a little bit more concrete question. Just hoping those of you with some involvement in the area can talk a little about how this sort of data, these sorts of designs are currently being used within drug development. I know I think there's a couple of you have some relevant experience. Pallavi, maybe do you want to start with some of the FDA perspective on that?
Sure. I can't say that I've seen this type of design in particular, where previous clinical trials, whether as a single trial or as a synthetic control arm, has come in for us to review at this time. But I do think that we have seen some sponsors and some applicants coming in with data from other external resources, external registry data, external EHR data, and have come in and asked us to review it. I think it's something that is discussed on a case-by-case basis, depending on the disease, and it's depending on the characteristics of a treatment, of a population, of a disease. It's something that we have interest in, but I'm not sure that we've seen this particular type of big design yet.
Everybody's looking at me. So we, as Pallavi mentioned, we have not seen data simply to replace totally like this using a prior clinical trial data. But we have had applications where sponsors have submitted to give a benchmark or a background rate to say that they are better than what is already out there. And the sources of data may be case reports or literature or registry data or real-world data. So we have seen all of this.
Ruthie, sounds like you have some experience with more early phase studies and using this sort of data there.
So Medidata has worked with a pharma company who is using the synthetic control arm approach to better understand their early phase studies, particularly one in AML, where their comparisons to historical literature sort of made them think that the experimental product was helping, but they weren't confident in that decision. And so we created the synthetic control arm to understand whether the results they were seeing really were improvement, helped them make the decision to carry forward a product further in development. And then we've also seen customers using this in late stages of development to monitor adverse event rates. So using the historical data to benchmark what is the expected rate of particular adverse events in the precise population of interest to understand whether their ongoing trials or even their post-marketing experience is sort of going well or in line.
So now, anyone else want to react to that? So I'll turn now to a question that I'm hoping that Antoine, Juhi, and Andrea can respond to, which is what do these types of analyses and studies mean for patients, for drug sponsors, and for regulatory agencies? So each of you gives one of those perspectives. So if you could talk a little bit more about how you view these sorts of studies and designs fitting in. Maybe Andrea, we can start with you.
Yeah, I don't think there's any great surprise. I mean, from a patient perspective, if you're going to enroll in a clinical trial, the hope is that you will be on the active arm, and so that you will have access to the latest therapies, the latest scientific knowledge, and so forth. So from a patient perspective, the more that we can enable that to happen, I think the better. But I think it's not just the clinical trial experience itself, as much as the speed with which we can, and the efficiencies that we can create in a full clinical trial sort of mechanism and paradigm, that it can help to hopefully solve for it and make it better.
Yeah, I think it really can be sort of that bridge between an imperfect data that might come from a single arm study where you're not really sure what you're comparing it with versus a randomized controlled trial. So sort of that in between to sort of bridge what you're finding. I think in terms of the augmenting idea, certainly could be used also as supportive. It's a different kind of augmenting, rather than just use as statement as a synthetic control, but to also potentially do this experiment and present it as additional evidence in conjunction with potentially a single randomized study or a smaller study that might be done. So I think these are areas where it can be used, and it would be helpful for us as a regulatory agency to have some kind of a little bit more standardized benchmark, because that's really, I think the most value in this is really how standardized it is versus looking at papers or historical data.
Okay, first I'd like to go back to a prior question, which is, has it been used? And I do remember in the early 2000s using taxotere head and neck phase 2 trying to submit an application on the basis of bootstrapping, because at that time there was no propensity score, but actually bootstrapping. Phase 3 were ongoing. The effector drug was so compelling that there was evidence, and the European and the US agencies did not accept this phase 3 based approach, and then ultimately what really struck me is that the phase 3 was exactly on the market what had been predicted. So I guess this is where my interest for this kind of approach. So it has been attempted for many, many years, out of recognition that the early knowledge actually can be more right than we think it is, absent of a prospective randomized trial. But back to your point, which is, well, from a patient's perspective, I do remember also another application for sunitinib where on the basis of the accelerated approval, the FDA notified officially from the IND that it was unethical to conduct a phase 3 trial in advanced EGFR lung cancer, just because of the magnitude of effect. And we had to convince the agency that, and brought Pasijane and others, to convince that it was necessary for the purpose of the rest of the world and the payers to actually conduct a randomized trial and expose patients to chemotherapy versus a very active agent, TKI, just for once and for all, really prospective. So this is very frustrating to be here, and to be forced to have no tools other than these attempts or this necessary conduct of the trial. I think what we agencies are most concerned with, we industry are most concerned with is what it is that the agency will be willing to say and do with this data, and what it is that it will mean, how it will translate itself into label, into summary basis for approval, what will the payer in the world will consider of this, and that's what we would like to work on, how we prospectively define what augmentation means.
Great, thank you. Just a heads up, I'm going to ask the panel one more question, and then we'll turn to some audience questions, so start thinking about what you might ask. So I wanted to turn maybe a little bit to the nitty-gritty, but to talk about some of the both challenges and feasibility of accessing and analyzing these types of data. So on some level, doing this successfully depends on the statistical methods, it depends on the disease context, but it also depends on just getting the data and having data that actually can be analyzed in these ways. Ruthie, maybe you could start with that.
Yeah, so when I showed you the presentation, I showed you the schematic of the dots and the circles, and I said, and then we curate and standardize the data, and the boundaries of those trials go away. And it sounded very nice, but really that was months and months of work, and there was a whole team working on that, making sure that the things that were measured in one trial were the same as they were measured in another trial, and if they weren't the same, deciding how we would reach a common ground and make them seem the same, and standardize all of those endpoints across trials. So this is part of the challenge. It takes significant resources to do this, but well worth the effort if we're able to.
So absolutely, that's historic data. But I think as we're having these conversations, we also need to think prospectively, and how do we do things differently to standardize inclusion, exclusion, what's being measured, how it's being measured going forward, so that as there is this emphasis on real-world evidence and so forth, we can set up the system now for what, based on all the pain that's been incurred to try to do the historic for the going forward. So I think that that's not something to be overlooked. It's something that really needs some significant time and investment.
Thanks, that's a great point.
So I think, one thing is about the follow-up is important to, as we are looking at the historical data, and particularly if we are looking at prior trials, if they have been stopped at an interim analysis because of efficacy or futility, either of those, you may not have all of the data that you would need to use it as a synthetic control. So if, going on what Andrea was talking about, if there is a push for further follow-up, even if at interim we decide that the drug is effective and we have complete data, that would be.
Maybe I can throw something out. I don't think FDA currently has any legal authority to use existing data for that purpose. I do know that FDA is the only place in the world which has a complete data set in terms of historical data for clinical trials. I mean, Medidata is a great company. You can probably have half of the market in terms of electronic data capture, so you have a great deal of data. We are far from having all. I don't know if the FDA has the legal authority, the willingness and the time to actually prospectively work with sponsors to define what will be, or would be, the future of randomized trial. But maybe there's something we should explore to see augmenting trial for the purpose of, say, we define the approval on the basis of approximate endpoints, which is validated, so it's clinically meaningful, but we still are interested in what is the survival. We're willing. Could we work together prospectively to define that you guys work with your data set, we provide the prospective data, we randomize 5 to 1, whatever, and then augment it. That would require probably some proactive collaboration work, which currently I don't know if there's any avenue for that, but I'm just throwing this out of the blue.
So I don't know that we would ever be able to share the data, but
no.
In a way, I think what you're suggesting is if you were to bring us a 5 to 1 randomization,
Yes.
then we would use the data that we have as comparison. But that's a risky endeavor because you don't know what's going to happen after using that data. So
No, we should not share that, and would you be prepared to put this in the label? I mean, if there are avenues, which are, why not?
I think I would put that.
yeah.
No, but it was probably a question too. No, but I'm just throwing out this. Unless we think bold, we will not resolve by just being cautious. We then find all reasons why it is difficult and we have to think it dirty. So problem solving is a real problem for patients and I'm just throwing it. Sorry for challenge.
In the reduce, reuse, recycle theme, then certainly trying to take best advantage of this today would be great. So this is a perfect opportunity. We're going to open it up to the audience.
So Pastor will first.
I think there's a difference between bold and reckless. Okay, and I don't want to preside over the death of the randomized trial here. Okay, and I think we would have to get. We have several statisticians here, but this needs further discussion in the statistical community, believe me, before we say we're not going to be doing any more randomized trials. To quote somebody that's not here, everybody's friend Tom Fleming, why do we do randomization? To take into account the prognostic factors we don't know about. Okay, and I don't don't think we should lose sight of that issue. And I'm not saying that this is not appropriate for certain circumstances as outlined here. Okay, but here again, I wouldn't want to say that, well, we're not going to do randomized trials anymore. Okay, and even though we give these special scenarios, having been in the agency now 20 years, I could see how our discussions with sponsors morph over a period of time. We have, you know, these now very defined situations. Well, why not move the envelope a bit more aggressively? Why not move it? And then eventually we'll see we're not doing randomized trials anymore. And I think that would be a difficult situation for anyone to suggest here. So I think there is an area of looking at this, and Raji is completely right. We do take single-arm trials. We have to be realistic. We've just written about this where we won't do randomization or where randomization might be difficult. But to just say we're never going to do a randomized trial anymore is something that I think we really would have to have further discussion about. But I'm not opposed to this. And the labeling would reflect what the data we used, obviously. If we're using external data, then that data would be somehow described in the label. But the other issue is who's going to be doing this synthetic control arm. You know, the sponsor, after they've looked at the data, really? Okay. Really? Okay.
I was not suggesting to replace, I was just suggesting trying to address a very concrete recent example with APA told the sponsor at the time I still think I would work with that you cannot conduct a randomized trial for a reason. And we essentially went back and convinced, yes, we have to and we will. So that you have that space where it's, I mean, the equipoise is lost or almost lost.
Well, I question, you know, how often we have equipoise in oncology because we do have preceding information that comes in. And equipoise is in the eyes of the beholder. I'm sure most of the pharmaceutical companies believe that they don't have equipoise when they're beginning the randomized trial because they already know in their hearts what they think is going to be the outcome.
This is a great discussion. I'm going to take moderator's prerogative, but while the mic travels, I'll take moderator's prerogative to say a couple things. Okay, over there in the corner. I think, right, no one, or at least I certainly, as a statistician, would never say we should not ever do randomized trials. I think one of the conversations for today is sort of where are the lanes where it really makes sense to consider these sorts of approaches. The other just sort of quick anecdote related to the point of sort of sponsors seeing the data. One benefit of propensity score methods actually is that you don't use the outcome at all. So personally, I have a little idea for if I was more entrepreneurial, I think someone could set up a company, like a CRO type company, to do propensity scores where they would never see the outcome. So they would be sort of the unbiased outside group that could do all of the matching. If they had access to the control data, they could do all of this and sort of have a firewall between that process and the actual outcome data. Again, that's my, if I was more entrepreneurial, I would have gone off and made millions of dollars. Instead, I'm a faculty member. So anyway, I think these are the sorts of things that really need a lot more discussion. So I think the mic moved over there. Great. Please just introduce yourself briefly and then.
Yes. My name is Catherine Lang from Pfizer at the head of data science and Pfizer.
Can you move the mic and make the mic higher?
Micro is not working.
I don't think it's working.
Hi, I'm Catherine Lang from Pfizer Oncology. I'm the head of data science. So two things. One's a comment and then one's a question. One of the comments is we talked about temporal controlling earlier on. In the European submission of our acute lymphoblastic leukemia drug, we actually found that temporal control was one of the most important things we could do in trying to find external matches because hematology care is so influenced by standard of care in support of therapies. Is that better? So in support, and we actually did a systematic literature review to decide what that temporal control would be. So I think it's almost reductive for us to say we can just pick a time and say that this is an appropriate temporal control. We did a lot of work to prove what that time was, and I think the Eli Lilly example is possibly
a criticism of just being able to use all of the data for the last 30 years and say this is an appropriate control. We should be defining that, and that's behooving to us as pharma companies and sponsors. My second one is a question really. Have you synthetic and external control arm as if you have within your group defined that for yourselves? I find that we flick in and out. I've just heard Dr. Pazda flick to one of them. Can you define it for me so that I'm going to be using it in the same way as you when I talk to you later on?
Yeah, I think that's a great question, and one of these examples where there's a lot of, from a statistical perspective, honestly, there might not even be a difference. And so then, is there really a meaning to it? So maybe...
So I can address that just from the point of view of my talk. I used external controls to mean any sort of external data, and in this case, it was data from a single clinical trial arm, the experimental arm of the previous clinical trial. And synthetic control arm, to me, and how I believe it was defined by this working group, is using the data from several clinical trials to combine them to create one common control arm to be used as a synthetic control arm.
Right, I agree. That is
but in all honesty, we could call everything as external control data. And Dr. Pazda says synthetic is something like a synthetic material of your clothing or something like that. So it is.
I do see though too that some people will use the term synthetic control arm when they refer to even real world data and utilizing that external control as a control arm. So I think we have to be careful. I think there is a distinction between what we're talking about today, utilizing the clinical trials data versus utilizing the real world data. So
I completely agree.
Control arm and synthetic perspective.
So I think the distinguishing feature at least for me, when I use the word synthetic control arm, that means there's been a matching approach applied. So we're not working with this large pool of all available historical data and we don't know much about it. We're working with a very precise set of historical data where we understand the baseline characteristics of those patients and we know they match the set of experimental patients that we're interested in.
In this kind of nascent field, I think it would be helpful for us to define even those things so we're all talking to the same language, and that would be very, very helpful even from this group as some sort of amendment to the white paper already.
It's a great suggestion. I have a comment as the microphone moves. Okay, I'll just add one quick thing sort of on that note. I personally, actually, in my own work, try to reserve the term control for a randomized setting, so say a randomized control, and I try to use comparison for non-randomized, so sort of a comparison group design versus a historical control design. It's just a minor terminology difference, but I think partly sometimes the terms are important because you sort of, you want to distinguish a randomized control arm from, say, a comparison arm. So I think all of that is to say that the terminology is really important and deserves a lot more care. Over here, thank you.
I might also the microphone is not working.
I think we can hear.
Okay, so I'm Nancy Valenti from Genentech, and I'm just really intrigued with the comment somebody made about the FDA holding the mega database of all the clinical trials that have been conducted and submitted to them. And given the conversation we're just having on the heterogeneity of the definition of these various types of data, is it real world data from community sites? Is it from clinical trials and metadata, our own clinical trials? I think all the companies and various people have been trying to figure out how to put this together and use it to accelerate development. I'm wondering if we could do something in partnership with the FDA where instead of the burden being 100% on the FDA, instead of the burden being 100% on the FDA to do these kind of analysis, we could do it in partnership. It's what you mentioned, this idea of having the CRO, and we could do it in a way with the firewall, as you mentioned, where the data is de-identified, anonymized, defining periods of time that would be appropriate to account for the drift that Raji talked about. I think there's a lot we can do, and we can come together as a group.
Anyone want to react to that idea? Provocative?
I mean, it's much better than that's what I was trying to allude to. Is that there's one place to go where there's a unique data collection that currently is untapped over them looking at the VMware affiliation of 2020 and 2030. And how can we essentially use that? In very specific circumstances, it's not a death randomized trial, but in very specific circumstances, why don't we try also to use that? That was my...
I just want to add that perhaps in other European countries, or at least in some of them, they have full patient collection of data. They have a complete registry of data where they can collect, at least in some countries.
Yes and no. I mean, I know that very well. I mean, the Nordic countries have very good registry, but they are registry very minimal amount of information. At baseline, they have no information, no annotation in terms of treatment. And then they have the death outcome. And then you have France, which is essentially Jean-Charles Soria is here, the contributors including that. France is probably the last place with unique answer where you have a national registry which accounts for 40% of patients, which are better than any electronic data record in the US or anywhere else in the world. But which really goes back to where are these data? These data are, maybe data has half of the clinical trial data in modern time. The EMD has probably all the data which have been the basis for approval for all recent drugs. And everything else is fragmented. So back to my comment from Genentech, commenting, I think there is really opportunity if we really try to crack open that problem off. In very specific cases, we have to randomize trial. How do we do this? And it's obvious that we have data. So.
Any other quick reactions to the feasibility? But yeah, as a metadata perspective.
Sure. Yeah, I mean, from a metadata perspective, we maybe are in a unique position in that we have access to such a large volume of clinical trials, about 15,000 trials in our database now. But I do believe there's potential for other data sharing initiatives, or even large companies or CROs that they have to establish the procedures to make this happen, but they have the potential to have a resource like this as well. And so I think all parties involved are going to have to contribute. I do think the role of the FDA is a special one. I mentioned in the beginning too that I have worked at the FDA, so just so that you'll understand the context of these comments. The resources that are available there to do this kind of work may not be sufficient for what we need. And also, maybe more importantly, the FDA supplies a critical review of data that is an important process, part of the process. To ask them to first create the scientific argument and then secondly evaluate it for approval seems like a little bit of a contradiction there. So I do think we need the FDA to work with us to make these things happen, but to put it completely on the shoulders of the FDA is probably not the right solution. Can I add one more?
Yeah, and then we're going to try to do 2 more questions.
I want to respond to Dr. Pasteur's suggestion that Tom Fleming says randomization is to balance the unknown covariates. And you're right, he's right, that's what it's for. And we don't have that with synthetic control arms or with these matching approaches. But what we've talked about with this working group in looking at our future endeavor, and this is part of what I suggested at the end, where we want to see if the difference between the synthetic control arm and the experimentally treated patients can replicate the difference between the randomized control and the experimentally treated patients. If we see that successful, then we can inch away at that to say, what if there's unobserved characteristics here that are imbalanced, and they're imbalanced to a certain degree? Would that have changed our overall conclusion about efficacy? When we get to the point where we can make those tipping point arguments, I think we can answer your question about unobserved characteristics. It's a really important question.
Thanks for bringing up that point. So we're going to do 2 questions. We've got one here and then one in the back corner. So we'll start.
Yes, I'm Lola Fisher-Najee, and I just, as a follow-up to the 2 case presentations, which I thought were really great, but I think the issue of temporality, which is one that's come up in a couple of questions here, is one that is what gives us a lot of pause with the use of this kind of methodology. And I'm wondering whether why we didn't, in the case studies, include sort of a determination as to the supportive care that were offered in the trials as a way to sort of match patients to get much closer to your sort of contemporaneous trial.
this is something we considered for this case study, and it's mostly a function of the data standardization process. So the form in which those kinds of things are measured in historical trials varies greatly from one to the next. Some trials measure number of previous types of one kind of therapy, others measure is this your first presentation of the disease? It's just really hard to congeal those data. And yet what I want to point out to you is that imbalance that we saw between our historical data and our target before matching, that went away when we did the matching. And our theory about that is that the characteristics that we did use are correlated with the passage of time. And so by adjusting for those baseline characteristics, we in essence adjusted for the changing standard of care over time by using those. But I do agree with you that if you had available something like the calendar time of when the subject was enrolled, it would be a great asset to use that in your matching process.
I think we have to be careful in some of these, yes, baseline matching we can do, but particularly if we go to hematologic malignancies where there can be transplant and all of those, then it does get complicated and it does vary from practice to practice or trial to trial.
That's actually what I wanted to mention also. That's a really great question. It's not quite as relevant for my case study only because there were only 2 studies that were being compared and we kind of understand them a little better, and I mentioned that that might have affected some of the differences that we saw in overall survival. But propensity scores can only take into account information that's pre-treatment, so baseline characteristics. Anything that you believe may be affecting patient survival or progression that may occur post-randomization or post-initiation of therapy isn't something that can be taken care of by propensity score matching. Now, there are other statistical methods that can be considered, but as you layer statistical methods on each other, the interpretation of your results also becomes more, for lack of a better word, convoluted. You're now narrowing the scope of what you can say about your results because of those different methods. But certainly, I do agree with you that even if you just were to look at the temporality of when the patient was randomized to a trial, the date of randomization, that is a baseline characteristic that might give us a lot of information. In this particular case, the first case study was able to match the patients very well, but maybe in other case studies that might end up being much more important.
Great, so we'll have one final question in the back.
Jeff Oxnard. I'm an investigator at Dana-Farber. So a question about whether the FDA data set is rich enough to serve as this historical comparator. One interesting element of the FDA data set is that the follow-up is not complete. The data is submitted and if there's a label, the follow-up stops. And yet we've now recently seen 3-year, 5-year follow-ups from some remarkable trials where the sponsor continues to collect data, but that data does not become part of the FDA data set. And so is it possible that in fact the data set might be too limited to serve as a comparator for these kinds of analyses? Would it need updated data that the sponsors actually hold in order to make these kinds of analyses possible?
Very good point. And yes, our data is limited. It's not only limited by the follow-up data that's not there in the products that are approved, but we don't have the trials where they didn't succeed or they didn't come to the agency for approval. So there are many more trials outside which we don't have any data at all. So no, we don't hold all of the data.
And Antoine, would sponsors want to submit updated data to the FDA to flesh out the data set, do you think?
I think unless we ask ourselves this question, we will not resolve the problem. So I think it's more interesting to try to identify what it is that will help us address the problem, as opposed to identify all the reasons why we can't. So I think that's the whole purpose of that workshop, is to really move forward. We don't have this answer. And the short answer, Jeff, to your question, yes, we would be willing, at least I would be willing to submit data if it contributes to greater science and better benefit for the patient. I would definitely be able. And really,
yeah, I agree. I completely here. I think that the spirit of the conversation here is the right one. We do all have to work together to make this happen. We do have to find ways to share this data across companies, across the industry really, and find a way to put this together so that the FDA can evaluate it.
use the last few minutes just to see, maybe go down the panel row and see if, let the panelists have sort of the last word. If anyone has a final thought that they want to do. Roger, maybe again, let's start down at your end.
I thought you were going to start from
well, yeah.
I think, as I said, certainly using external control data in places where today we are accepting single-arm studies is certainly better than single-arm studies. But we should not sort of say this is going to be the new standard and don't think about randomized controlled studies. For example, in adjuvant studies, we certainly would want to look at randomized clinical trials. But there is hope, and this type of analysis, aggregation of data, if nothing else, it helps us understand the disease as well as what's available at this time, better than nothing.
No, I just want to, I'm very encouraged and energized actually by this conversation. And I have been by the work, and very thankful for Fund Cancer Research for essentially providing the framework for this conversation and the drive. We have an obligation as partner to advance the science to the benefit of patients. Not any one of us can actually achieve the whole journey. And it's only through the partnership between the payers, the regulators, the scientific community, the academic world, the industry, and the regulators that we will be able to advance this in that space where we currently are so short, where we can't, we are the margin of not being able to randomize patients. At the same time, we want to have this data. So I'm very energized by this workshop. Thank you.
Great. Judy? So as I said earlier, I really wasn't sure what to expect in terms of the results going into this, and had some experiences with some brain tumor clinical trials that were randomized where the result actually came out unexpectedly. And it's only because there was a randomized arm that we saw what the true treatment effect was. And in a situation where if they used well-established historical controls, we might have made a decision in a different direction or an assumption in a different direction. So I was curious to see what these results would be, and it also, I think, is very energizing as well. It's sort of inspiring to go back and see if we can look at our databases to see if I can answer some questions about some of the other results we've seen in these trials.
I too, I share your optimism. I think, I don't think I'm quite as impatient as Ellen, but I think I'm pretty close to it in terms of converting things into action. And so I think that it's fantastic that we're looking at this, that people are demonstrating that you can use the data, figure out how to clean the data, identifying the situations in which it's appropriate, and those in which it's not, because it might not be appropriate at all. But I look forward to having a meeting maybe next year or the following year where it's not looking and proving that we can do it, but rather case studies of where it's been done to demonstrate that it works.
Thank you. I think this was a really interesting discussion, and I think that the two case studies really did show some promising results. And while I do agree with Raji that we should proceed with caution, I think that means that we should proceed. But we should also proceed with an eye on other areas where this type of data can be used. I think that there are a lot of low-hanging fruit, if you will, where we can really utilize this type of data and do something very good with it, rather than just focusing on replacing or augmenting clinical trial arms while we work towards that goal as well.
Again, I'm really fortunate to be sitting at this end because I'll agree with what everyone else said. I'll just add a little bit about the patient perspective. Not that I'm an authority on this perspective, but just because it hasn't been said yet, which is that anything we can do to first increase the chance that patients will be assigned to the active arm, and second, to speed the development of good products. That's something we should invest in. I think this is that type of thing. So I'm like Antoine, I'm really energized by this conversation today, and I hope we will be here next year talking about examples that have been successful.
Great. Thank you. This is a really great discussion. I think some nice concrete steps for moving forward and things to think about. So let's thank the panel.